Hello. Okay, hello everyone. I think it is about time we start today's webinar. If you have any questions, problems connecting, please use the chat and you can talk to us through the webinar. And I'll start here, introduce our speaker for today, and then he'll start his talk right after. And then we'll wait for all the questions at the end. So the Early Career Member Network always organizes these seminars once a month. This is the 4th session for this year, and I'm Fatima. I work at the University of Nebraska-Lincoln. I'm a committee member. If you have any suggestions or so on, you can use our email address that's at the bottom of this page, info.emn@themetabomicsociety.org, to reach out to us. You can also connect with us online using any of the social media platforms, Facebook, Twitter, LinkedIn, or email. And you can send us any suggestions, recommendations that you might have or that you'd like to see the Early Career Member Network do for you as members of the Metabolomics Society. With that being said, the Metabolomics Society's conference is going to be in The Hague, Netherlands this year, starting June 23rd. The registration deadline is actually today, and EMN will also be represented there. We will have a reception and a career night organized that's part of the schedule, and we're actively planning that. So if you would like anything included as part of the career night or the reception, please feel free to send us your suggestions. With that being said, I'll introduce our speaker for today. He is Dr. Robert Powers. He is a professor of chemistry at the University of Nebraska-Lincoln and also the director of the Systems Biology Core at the Nebraska Center for Integrated Biomolecular Communication. Over the years, Dr. Powers has worked in both the industry and academia, starting with the industry and working at Wyeth for over 13 years before moving to the University of Nebraska-Lincoln. He has numerous international and national patents and has worked in across different disciplines in NMR spectroscopy, and now is moving towards combining NMR and mass spec metabolomics work to study various diseases. Today, he's going to talk about combining NMR and mass spec metabolomics to study different neurodegenerative diseases. With that being said, and that short introduction, I will now pass over the screen and the audio to Dr. Powers so he can start his webinar.
Well, hello, everybody. I'm Fatima. Thank you very much for the invitation and that very nice introduction. And thank you for the opportunity for me to talk about our recent work in using both an NMR and mass spec approach to metabolomics and applying that to a number of ongoing studies involving neurodegenerative diseases. I'd just like to start off with a general overview of my talk. I'm going to divide it into effectively 3 sections. Talk about the application of NMR and mass spec to metabolomics, and hitting a particular areas related to expanding the coverage of metabolome, some common sources of errors and misunderstanding applying metabolomics and data analysis, and some issues in terms of trying to improve our efforts in terms of data collection and processing. I'll then turn the attention to 2 projects, our application of metabolomics to Parkinson's disease, and there'll be 2 subcategories looking at the molecular mechanisms associated with paraquat, and then the synergistic effect of both paraquat and alpha-synuclein. And then I'll turn to another project discussing our efforts to diagnose and treat multiple sclerosis, and I'll end with a brief conclusion. So as most of you know, metabolomics is experiencing a tremendous exponential growth. And this is potentially reason because metabolomics can be applied to a whole variety of different fields, from personalized medicine, drug discovery, agriculture, nutrition, and so forth. Also, it's a relatively universal methodology that can be applied to a whole variety of different types of biological cell samples. In fact, our group has worked with almost all of these areas, from biofluids to tissues to cell extracts and so forth. And as a result of this, another reason why metabolomics is also experiencing this growth is that there's a relatively low barrier to entry. At some levels, metabolomics is viewed as relatively simple in principle. The main idea behind metabolomics, as most of you are well aware of, we're going to go ahead and collect multiple biological samples in order to go ahead and try to characterize the state of the system, right? See what metabolites pathway differ between a disease state and healthy individuals, or between a genetic mutation. And then we're going to go ahead and apply some basic procedures to extract and pull out the metabolites and remove all of the biomolecules, tissue material, and so forth, and then characterize the chemical signature by using NMR or mass spec, and then apply some advanced statistical approaches, both multivariate, univariate, to see whether or not we can identify what the actual differences are between those particular systems. And again, the idea is that we can feed into these statistical analysis mass spec data and NMR data, and then hopefully we're going to see in terms of our multivariate analysis, our score plots, do we see distinct clustering? Do we see differences in terms of the underlying spectral data indicating that there's a metabolomics difference? And if so, then we can go ahead and apply other statistical means to identify what the actual sources of those differences are, identify the associated metabolites, correlate that to the actual networks in terms of metabolic processes, to get some insights in terms of what other fundamental differences between the systems that we're actually studying. But fundamental to this entire analysis is that the assumption is at the end, when we're looking at the statistical data and results, and we're looking at the resulting NMR mass spec data, that it's reflecting the true metabolome, that these changes are actually biologically relevant. And in the case of metabolomics, this is a really serious concern. So right now, metabolomics does face a number of technical challenges associated with getting reliable, reproducible biological insight in terms of the system. Probably one of the biggest challenges that we face is that the metabolome itself is inherently very unstable. Metabolites undergo chemical changes relatively rapidly, can get oxidized, differences in terms of stability, and this is all going to be reflected in terms of how the samples are handled and processed. Also, there are common issues and problems with the applications of these advanced statistical techniques. A lot of people who are involved with doing metabolomics are not necessarily statisticians or having a strong statistical backgrounds, and these approaches can routinely be incorrectly applied, which lead to inappropriate biological inference. Also, the process of identifying and detecting all the metabolites is extremely challenging and difficult, and I'll address this more. And the field is still relatively new and evolving, so standardized protocols are still lacking. So if we think about one of the major issues that we're dealing with in terms of metabolome, again, the metabolome itself is extremely sensitive. Different metabolites have different relative stability, relative turnover rates. The metabolites itself can change within millisecond time frames. And we look at stability in terms of a sample sitting on the desk with over 24-hour period. We're going to see some metabolites are going to be significantly lost or degraded, while other metabolites are relatively stable. So various aspects in terms of how the actual sample is prepared, removing enzymatic activities, how the samples are collected, processing, are all going to contribute in terms of what metabolites are still available and still detectable and at their relative particular concentration. And again, these are things that are not related to the actual biological system that's being studied. So again, if we think about all the steps, all the processes that we need to do in order to properly do a metabolomics experiment, there are sources of errors at each of these steps. How the sample is prepared, how the data is acquired and processed, how statistical and methods are being applied, what source of the data that we're actually getting, who is handling that. And all these issues can lead to erroneous results if we're not aware of this or we don't deal with these in terms of compensating for this. So to illustrate this, I'm going to shed some of our own dirty laundry to illustrate the fact of how, this sensitivity in terms of metabolome can lead to kind of false results. So here's some data being processed by a single student. They're working with analyzing, preparing the samples in the morning, decided to take a break for lunch and come back in the afternoon. And the resultant statistical analysis shows that the clustering is based on which samples were prepared in the morning versus the afternoon, as opposed to the actual differences between the drug treatment that we're interested in. Again, here's another subtle example where two students are working side by side, following the exact same procedure and protocols, but the separation that we see in terms of the PCA scores plot is based on which student handled which sample, again, instead of the desired underlying biological in terms of differences. So again, we need to appreciate the fact that, again, the metabolome is very sensitive, and anything that we can do can impact an unintended bias that can give us a false impression in terms of what's going on. Again, the statistics that we can apply can also lead to very misleading interpretation. I think Mark Twain's quote is something that we should very adhere to, that there's lies, damn lies, and statistics. So for instance, the multivariate techniques that we commonly use are designed to find differences, to the extent that even if you go ahead and feed in random noise as shown here in terms of this PCA scores plot, we can get excellent group separation, even though this is simply random noise. We also recently demonstrated the same impact by looking at real metabolomics data, where we can get very nice class separation using both PCA and OPLST data. And then we systematically added noise to the actual original data. And the PCA scores plots behave as you expect. As we increase the amount of noise, the group separation diminishes, the variance in terms of the group increases, and eventually we have a random scattering. But when you're dealing with supervised techniques like OPLS, again, you're defining group separation, and OPLS gives you what you're asking it to give, and you get perfect separation, even though the PCS tells us this is primarily random noise. And what's scary about this result is that if we go ahead and look at the correlation between the unloading loadings, or the key features that are actually defining this group separation, and without the noise and comparing with the noise, we see that there's absolutely zero correlation. So while you can go ahead and interpret these scores plot and identifying underlying metabolites that define these differences, they have absolutely nothing to do with what the actual true differences were. So multivariate techniques require a very strong, valid approach to validate the data. And one of those approaches that are commonly used in terms of reporting R-squared or Q-squared values. Well, these are values that are actually important to interpret the quality of the actual model, where R-squared does provide a measure of the actual model fits, and Q-squared kind of gives us a measure of consistency. R-squared and Q-squared really don't give us any measure in terms of the validity or the reliability in terms of the actual model. And again, if we look at the example that I illustrated where we're increasing noise, where we saw no clustering differences in terms of PCA, but the OPLS gave us nice separation, the R-squared and Q-squared actually fall into values that are acceptable. Right? Both are significantly greater than 0.4, and there's not a tremendous difference between R-squared and Q-squared, but if we validate this model using CV ANOVA, we see that this model is actually false. So R-squared and Q-squared simply by themselves do not tell us whether or not a model is valid. Another concern in terms of applying statistics to metabolomics data is the routine application of a simple pairwise comparisons like students' t-test. Well, these are very valuable and valid in terms of when you're looking at a pairwise comparison. The important difference in terms of with metabolomics or other omics techniques is that we're actually looking at a collection or a large number of metabolites that might differ. And it's important to appreciate that error is additive. So while maybe any particular metabolite might be statistically significant, whether the student T-wit test or comparable in terms of the pairwise comparison, the entire set of metabolites might actually be deemed in terms of being statistically insignificant. Again, the error is cumulative. So we take a simple example. If we have a set of 13 metabolites, and each one of those gives us a statistical significance at a 0.05 level, the actual significance of the entire set is insignificant, giving us a p-value of 0.49 because all those individual errors increase. Now, this doesn't tell us which specific metabolite is erroneous. It just simply tells us that there's almost a guaranteed level that at least one, if more of these metabolites are no longer statistically significant in this set. And so again, accounting for this false discovery rates, applying something like a Bonferroni adjustment or Benjamin-Hosberg correction is necessary when we're actually doing metabolomics analysis of a large group sets. So again, this simply highlights the fact that the field needs standardized protocols, data processing, and model validations approaches. And one of the things that we've been working on in my group to address this is that we have developed our MVA-PAC chemometric software. This is freely available through our software. It's also available as part of NMR Box. The MVA-PAC provides a single processing step where raw data can go in. All the necessary processing, pre-processing, model generation can be generated. And we developed this initially for NMR, but we've also been incorporating capabilities to go ahead and do mass spec data. An important aspect in terms of MVA-PAC is that it includes, as kind of a standard part of the process, validation of the resulting model. It includes some standard permutation tests and calculations of CV and NOVA p-values. And again, a cross-validation permutation test shows us how we can go ahead and leverage these R-squared and Q-squared values to infer validity. If we know what the actual true values are from the model, we can go ahead and permutate the membership in terms of the groups, recalculate R-squares and Q-squares for all these false models, and hopefully we see a very large difference between the false models and the true models. And also we can get out of this, since we're doing these multiple model calculations, we can also get an associated error with each data point in our scores plots and telling us how reliable or how stable any particular data point is. And again, this is really nice data showing all the points having very little variation depending in terms of the permutational approach. And again, this is a standard output in terms of MVA-PAC. So with the availability in terms of MVA-PAC and a standard platform, we can now go ahead and start asking some fundamental questions. For instance, what we know right now is that in MVA-PAC, we have over 9 different normalization methods that are available to the actual user. And the question is, which one of these would be a preferred option? Which ones perform better or worse? Which one should be kind of the default choice? And in order to be able to address this properly, the best way to do this is dealing with simulated data so they actually know what the actual outcome was. So in this case, we took simulated NMR data, and then we modulated that data based on its dilution, relative intensity of the peaks, and also relative additive noise. And we simply just increased the amount of dilution, increased the amount of noise. And we can see that at relatively low noise, low dilutions, most of the normalization procedures work equally well. The lone exception is the histogram matching, which is one that I really caution you to avoid because it actually performed poorly even at relatively low noise and low dilution effects. But again, as we continue to stress to the data set, add more noise and all dilutions, we start seeing a separation where a couple of normalization approaches still do much far superior performance versus others. And from this analysis, what we see is that the constant sum, PQ, and region of interest all performed the best over a large range. The other thing that was really interesting or surprising from this result was that the realization that the dilution factor really had no impact in terms of the performance of the normalization. No matter how large a range of intensities, multiple orders of magnitude, all these normalization factors were able to go ahead and recover and regenerate the original data sets. So again, what this told us is that the major detriment to analyzing spectral data from metabolomics is noise. That as we increase noise, the performance of all of these normalization approaches diminish rapidly. And it also allowed us, we go ahead and take the best performing methods. We see that roughly when you get to around 20% noise or lower, that the methods perform well. But as soon as we get to that 20% noise region, we start seeing a fall off. So this again tells us now that when we're looking at the quality of the data we're collecting, clearly minimizing our noise is going to be a key factor determining whether or not we actually get a reliable results. Another side point in terms of that's particularly unique in terms of with NMR is that, again, NMR can induce false separations or false factors due to the fact that the NMR spectrum needs to be phased. So here's data, uncorrected data, that actually shows this minor phase corrections. These corrections are probably less than about a degree, but these variants can lead to false group separations. So we've developed an algorithm that simultaneously corrects to phase and also corrects the scatter or the dilution factor by fitting all the NMR data to an average norm. And this again shows you how well this particular works, going from the uncorrected spectra to the corrected. We can see we're getting now uniform intensities of all peaks, but more importantly, we're getting really beautiful phase correction. And again, this normalization technique, PSC, is involved in MBA-PAC. And then when it comes to NMR data, this outperforms all the other normalization techniques that I was discussing previously. Another question that comes up is what multivariate models should we actually use? Does it matter? Again, MBA-PAC contains 5 very common multivariate statistical techniques that are used by the community. And we wanted to, again, address the question, do any of these perform better or worse than the others? And again, the best way to go ahead and do this is with simulated NMR data, where we know actually what the differences are between the two sets. And so when we go ahead and generate a model and look at the loadings, we know exactly what peaks should be present because we have defined that in the actual spectral mixture. What's actually very comforting about this study, and this work is under review right now, is that when we're dealing with relatively modest noise and variations in terms of the data, that all the models perform equally well. We looked at 3 different classifications, how well it actually identifies group membership, looking at the area under the curve, and how well it produced the actual true loadings, again, since we know what they are in terms of this assimilated data set. But again, as we really try to stress this data, and we have to stress this to an extreme, and going to very small differences and large noise, only then do we start seeing some separation between the models. So again, what this tells us is that under most circumstances, any of those 5 models, LPLS, PLS, SVM, random forest, or PCA, LDA, are going to yield relatively comparable results. Only under extreme circumstances are you going to see a separation, and in those circumstances, we identified OPLS as actually performing the best. So as I indicated earlier, again, one of the challenges with metabolome is going ahead and detecting and identifying the metabolites in any particular study. As of now, the metabolome is vastly unknown. There's a large amount of dark matter, so to speak. There is a lot of estimates in terms of what the number of metabolites, approximate estimate in terms of human metabolites, including all sources, maybe upwards around 150,000. Plant metabolites might be as large as 1 million in terms of more. And if you think about it, the typical metabolomic studies, we're only seeing a small percentage of that, a few hundred metabolites at best. And I think part of this reason is that most of the studies, most of the community is relying on only one analytical method. And I'd argue that this undermines their ability to cover the entirety of the metabolome. That the 2 most common techniques we use is NMR and mass spec. And again, they're primarily used separately. And the reason is based on that is individuals' expertise or experience with the particular techniques. But NMR and mass spec are not equivalent. They have different strengths and weaknesses. And the best way to kind of look at this is that NMR it's going to be able to go ahead and detect the most abundant metabolites, whereas mass spec is going to detect the metabolites that readily ionize. And these two sets, as demonstrated by a few examples, are not necessarily the same. So by combining NMR and mass spec, you're going to get a broader coverage. And we demonstrated this in a recent paper where we're looking at the response to the metabolome of algae due to some drugs that were meant to improve fatty acid accumulation. We combine both NMR and mass spec, which required very minimal modifications to the actual sample preparation. Basically, the metabolome is extracted simultaneously, and then the sample is split for proper preparation for both NMR and mass spec collection. And again, very minor modifications to any standard process. And we can also go ahead and combine the NMR data sets into a unified single statistical model, as kind of demonstrated here in terms of comparing the metabolome response resulting from both NMR and mass spec data. And as a result of doing this, what we saw is that we got a broad, nearly complete coverage of all the metabolic processes that were responding to these particular drug treatments. What you're seeing here is all the pathways color-coded by whether or not their metabolites are detected by NMR in blue, mass spec in red, or both NMR and mass spec by black. And again, by combining NMR with mass spec, we effectively filled in a lot of missing holes that were only observed by mass spec. And in this particular example, we actually broke down where we saw roughly about 1/3 unique metabolites by NMR, 1/3 unique metabolites by GCMS, and 1/3 of those metabolites were detected by both NMR and mass spec. So again, without covering that, we wouldn't have gotten a true picture of the impact of these particular drugs. Now, one of the challenges is that, and again, NMR and mass spec are fundamentally different. And when it comes to their quantitation in terms of the absolute change of the metabolite, while we saw a general trend, there is still somewhat significant scatter. And in this case, NMR is a much better technique when it comes to quantitation when you're dealing with on-target metabolomics, where things have not been calibrated in terms of mass spec. So how are we able to go ahead and generate two very distinct data sets and combine them with a single statistical package? Well, we applied multi-block approach, both the PC and PLS, and again, these are all available in our MBA Pack software. And basically how these approaches work is that each individual data set goes into its own matrix. So the mass spec goes into one matrix, NMR goes into another matrix. We generate particular models in these individual matrix. And then the block scores from each matrix are now moved to a super level to generate a super block score. All right. Now, as I indicated, that OPLS seems to be the best performing technique, and there's also a lot of other advantages relative to PLS. The main important difference between OPLS and PLS is that PLS confounds in the model both the defined group separation and any confounding factors. So if you're thinking that this is maybe a study comparing healthy individuals versus individuals with various different diseases, you're going to see that these ellipses all are skewed along the diagonal, because the PLS model will incorporate both the differences between healthy and disease plus any other confounding factors, differences in diet, differences in age, differences in relative health level. OPLS solved this problem by forcing only the group separation and along the X diagonal, or TP, and moving all these other confounding factors into the Y dimension. And this is visualized by this, lack in terms of the actual tilt, but it's really more important in terms of analyzing the data that the difference between any particular groups now are going to be driven by that group definition, healthy versus disease. And so to address this, we also developed and derived our own multi-block OPLS model, which is implemented in terms of MBA Pack. So this is available for use that way. So again, giving that background, considering all those particular factors, developing protocols, applying statistical techniques, and a reliable and robust approach now gives us the confidence to actually apply these techniques to real biological systems and expect that the outcome is going to be of biological significance. So one of the projects that has been ongoing in my group is working in terms of Parkinson's disease. Most of you might be familiar with this disease. It's a progressive neurodegenerative disorder that leads to loss of control in terms of muscular function. Individuals suffering from Parkinson's disease tend to have tremors, difficulty walking, and other factors. The underlying cause of Parkinson's disease is the loss of the dopaminergic neurons in the substantia nigra. Those are the neurons that are responsible for producing dopamine, which is distributed to other parts of the brain, the cauda and putamen, which are involved in controlling muscle function. So currently there is no cure for Parkinson's disease or treatment to stop the progression. The exact cause of Parkinson's disease is unknown, but there's a growing appreciation that it's actually a multifactorial disease. The factors associated with aging, genetics, and importantly, environment all contribute to Parkinson's disease. In fact, only about 10% of all Parkinson's disease cases can be attributed to hereditary, and those are ones associated with very known mutations, particularly alpha-synuclein is a common mutation associated with Parkinson's disease. So when we're looking at sporadic Parkinson's disease, these genetic alterations are associated with some sort of environmental or occupational factor. And a number of environmental agents have been linked to Parkinson's disease. Pesticides, heavy metals, and so forth are the prime factors. So actually there was an extremely large epidemiology study done across the US looking at Parkinson's disease, looking at the prevalence of Parkinson's disease, and what was known is that in the Northeast and the Midwest is the largest Parkinson's disease. And we think about what that correlates with. It correlates with agriculture, the application in terms of herbicides and pesticides, or in the Northeast, it's associated with industrial sites, particularly associated with exposure to heavy metals, such in terms of manganese. So again, Parkinson is been associated with the pathology of Parkinson's disease, and it's inducing this dopaminergic degeneration. So another factor that's known associated with Parkinson's disease is that environmental toxins seem to lead to mitochondrial dysfunction. And again, this makes sense when you think that the neurons have a very high energy demand, which the mitochondria is going to go support. And it's a very high glucose usage. Again, glycolysis associated with mitochondrial activity. And again, over 25% of all glucose usage is being used by the brain. So again, mitochondrial dysfunction has been associated with Parkinson's disease. Toxins have been known to alter redox homeostasis, affect energy metabolism, perturb central carbon metabolism. So again, this gives us this clear entry that there's an important role for metabolomics for investigating Parkinson's disease. We also know that that's not the whole story with Parkinson's disease. Another hallmark of the disease is this formation of Lewy bodies in the neurons. These aggregates of protein and other material is a pathological hallmark of Parkinson's disease. And a major component of Lewy bodies are alpha-synuclein. And again, mutations in alpha-synuclein have been associated with particular Parkinson's disease. This protein has unknown function. Its structure has been solved. It's primarily a helical protein with a large component in terms of unstructured. And what's well known is that changes in environmental conditions can induce a conformational change in alpha-synuclein, result in terms of this oligomerization in terms of beta sheets, forming these amyloid fibrils, which are the function in terms of Lewy bodies. So again, there seems to be the connection between this formation and perturbation in terms of the metabolic homeostasis of the neuron. So again, to summarize, mitochondrial dysfunction and any energy failure are associated with environmental toxins. And this process can also seem to be associated with this propagation of the amyloid fibrils by the aggregation in terms of alpha-synuclein. This change in the environment both induces the formation and aggregation, and also decreases the ability to clear these aggregates, and so we get this accumulation leading towards the neuron cell death. So again, there seems to be this synergy between environment and genetic processes. So in our particular study, we're using both cell tissue cultures of dopaminergic neurons, and also looking at brain dissection from mice. And again, we're using a combined mass spec and NMR analysis. When we started on this particular project, again, mitochondrial dysfunction and energy failure was the key focal points. And again, we had this correlation of herbicides, pesticides, and also designer drugs with Parkinson's disease. In fact, there's a relatively large collection of drugs that are all known to induce cell death or diminish cell survival in terms of dopaminergic neurons. And again, as we started this process, these compounds were all treated as equal as a chemical probe of Parkinson's disease, because they all led to the same endpoint in terms of neurogenic death. But what we showed early on in terms of, we looked at the metabolic profile of these drugs, they all had very distinct impacts. So telling us that, well, the endpoint is the same, the underlying molecular mechanism was different. And we decided to focus on paraquats because it has a long-standing correlation and also one of the largest impacts in terms of metabolome. So what we also noticed early on that made paraquat relatively unique compared to these other treatments is that the impact in terms of paraquat was irreversible. That if you treated the cells with paraquats for about 24 hours, washed the paraquat away, the cells still died. Whereas if you go ahead and did that with other toxins, if you go ahead and look at treatment later on, we were able to go ahead and wash away the effect and only required a much longer treatment. So again, further illustrating this process, that this idea that the molecular mechanism of these treatments were not the same. As I said, we went ahead and combined both NMR and mass spec data to look at the metabolic changes in these dopaminergic neurons upon treatment in terms of paraquat. And again, this is just showing you how our multi-block approach, our MBA pack software can help facilitate the analysis, because what we can generate is a uniform model, but also individual models, and we can generate these backscale loadings from the NMR data and the mass spec data, and really telling us the major peaks and the corresponding the major peaks, the metabolites that are associated with the response in terms of paraquat. We can go ahead then and follow that up with other statistical analysis, looking at a standard S-plot, comparing the response in terms of paraquat. And what we see again is a large increase in terms of metabolites associated with glucose metabolism in the pentose phosphate pathway, in terms of both increasing, in terms of decreasing. And if we go ahead and do an SUS plot comparing paraquat to other drugs, again, we see these metabolic changes are unique to paraquat, again, illustrating the fact that while the endpoint is the same, the underlying molecular mechanism is not. Again, we can go ahead and compare the metabolome impact in terms of paraquat versus other toxins, looking at the changes in terms of intracellular metabolome and also the extracellular metabolome. And again, we see very distinct differences associated with paraquat relative to the other treatments. Again, that's consistent in terms of the univariate versus the multivariate analysis. We did not just simply do a metabolomics approach, we also combined proteomics under the same circumstances. And this also highlights, I think, the underlying challenge with proteomics is that we were able to observe a large number of changes in terms of the proteome, as resulting in terms of paraquat, number of proteins increasing, number of proteins decreasing. We can go ahead and identify that, but we get an extremely large list of proteins, and those fall across a whole variety in terms of different biological functions. And so actually be able to identify those proteins that are directly proximal to the activity in terms of paraquat versus those other proteins that are changing that are downstream, secondary, tertiary effects is very difficult to do. But the advantage of having both the metabolomics data and proteomics data is now we can go ahead and integrate those and look where they are overlapping. And where we see that overlap, the underlying assumption is that's probably where proximal to the actual activity in terms of paraquat. And when we do this, what we see is that we see an increase in pentose phosphate pathway enzymes and associated metabolites. We see increase in the PPP metabolites and associated enzymes. And again, we see decrease in glycolysis and TCA, right? And again, when we look at this individually, again, one of the advantages in terms of metabolomics, it's kind of hypothesis generating. So given this information, our collaborator then went back and said, can we identify any particular enzymes that are uniquely impacted by the addition in terms of paraquat? And what he observed was the particular enzyme glucose 6-phosphate dehydrogenase was G6PD was increasing in terms of its response to the treatment in terms of paraquat. So again, given that information, he went ahead and generates mutants that either overexpress C6PD or looking at cell lines where we knock down the activity of G6PD by the addition of inhibitors. And what we saw in both cases is that when we overexpress G6PD, we saw a greater response or greater sensitivity to paraquat, whereas there is actually no response to the other particular toxins. Again, suggesting that this is going to be their primary target. And even more exciting that when we knocked out or reduced the activity of G6PD by an inhibitor, those cells no longer responded to paraquat. They no longer died. We can dose them as much as we want, and we would still get cell survival. So putting this all together, combining the metabolomics work with the proteomics data and the more traditional cell biology, we were able to identify that paraquat hijacks the pentose phosphate pathway. Specifically, the actual target of this drug is G6PD. This enzyme is normally responsible for recycling NADP and NADPH, but paraquat can also undergo its own redox cycling. And again, what it's simply doing is using this regeneration in terms of NAD, NADPH as a source to undergo its own redox cycling and generating that reactive oxygen species, which is resulting in terms of leading towards the neuronal cell death. Right? Other aspects that we observed in terms of impact paraquat toxicity that again supports this particular model is that we observed that paraquat induces a glucose uptake. Again, feeding the this particular process in terms of this the pentose phosphate pathway. We also see that paraquat toxicity, right, is diminished. We can remove the impact of terms of paraquat if we actually inhibit this increase in terms of glucose transport. We can also remove paraquat toxicity if we grow the cells and glucose deprivation. And again, we see increase in terms of cell survivability. And again, we can remove paraquat toxicity if we actually inhibit the production, right? So again, this idea that from what the metabolomics is telling us in terms of the process, we can verify from a more traditional cell biology approach. Right? So again, this gives us the idea in terms of the response in terms of what paraquat is doing. It's inducing a larger impact in terms of the actual cell. It's increasing the amount of glucose that's being transported, feeding the glucose 6 enzyme is part of this PPP, which is resulting into ROS cycling. So not only is it impacting this activity, but it's also impacting other cellular process to again lead towards this paraquat toxicity. Right? Again, similar to this, right? Associated with this, we noticed that paraquat induces AMPK phosphorylation activation. And why this is particularly interesting, AMPK or adenosine monophosphate activated protein kinase is again, it's a master regulator of metabolism in response to energy deficiency. So again, we're seeing multiple symptoms as resulting in terms of paraquat activity that related to this toxicity. That not only is it inducing glycolysis, but it's also inducing the activation of the regulators in terms of glycolysis. So again, if we paraquat toxicity is increased with inhibition of AMPK. Paraquat and AMP inhibition impairs glycolysis and mitochondrial respiration. So again, we can get a synergistic effect in terms of the activity in terms of paraquat. Right? And again, paraquat toxicity increases due to AMP inhibition, which is reversed if we go ahead and actually grow the cells again in glucose deprivation. So again, giving us a further complete picture of the molecular impact in terms of paraquat. Now, of course, most of these studies were done in cell cultures. So it always raises the question in terms of whether or not this will be comparable if we're actually looking in brain tissues. So we also did a parallel mouse model where we treated mice, standard C57BL6 mice that are roughly 8 to 10 weeks old. We injected them with paraquat or salines, and then sacrificed the bright mice, removed the various different brain regions, and did an analysis in terms of the metabolome. And what we saw is that the midbrain had the largest impact in terms of metabolome resulting from paraquat treatment. And again, looking at the backscale loadings, we saw, again, from a validated OPLS model, again, we saw the same collection of metabolites and processes that were occurring in the actual cell cultures. So this gives us at least a level of confidence that what we're seeing in the cell culture is consistent with probably what's actually happening in the mouse, and hopefully the same thing in terms of human patients. As I said, Parkinson's disease is complicated. It's not simply a one factor, it's multifactorial, and genetics does play a part. And so therefore we went ahead and looked at the relationship or synergy between alpha-synuclein and paraquat toxicity. And as identified here, what we observed is that alpha-synuclein potentiates this toxicity and metabolic dysfunction. So again, if we go ahead and develop a cell line that overexpresses alpha-synuclein, we've shown this here. We have those particular cell lines. And if we go ahead and do that, as we increase the amount of alpha-synuclein, we diminish the actual cell survivability. And again, that impact is further exacerbated in terms of the treatment in terms of paraquat and also with a heavy metal. And if we go ahead and look at the metabolic change, comparing just overexpression of alpha-synuclein, overexpression in paraquat, and both the overexpression of alpha-synuclein and paraquat, we can see that there's a linear trend and there's a much greater impact by the combination of both alpha-synuclein and paraquats. And again, if we go ahead and look at the detailed analysis of the metabolic changes, we have the combined overexpression of alpha-synuclein and exposure to the paraquat, we see the same impact that we saw before with paraquat, is that everything is just greater. So we see a further increase in terms of glucose uptake and glycolysis. We're seeing to support that, a translocation of glucose transporters to the plasma membrane. And again, we see a greater increase in AMP activity. So everything that we saw with either with paraquat by itself is now just further emphasized in the presence in terms of the overexpression in terms of alpha-synuclein. And again, glucose plays a prime role in this, right? That we can, again, reverse the toxicity in terms of paraquat by glucose deprivation, looking at cells in terms of impact. And again, whether or not it's just paraquat by itself or combined with alpha-synuclein, we can go ahead and regain cell survivability. And similarly, again, we see the same synergistic effect, increased effect in terms of the metabolome response to AMP activation. And again, if we prevent AMP activation, we can also prevent paraquat-induced cell death in the presence in terms of paraquat. Again, so we're seeing this really nice across the board synergy in terms of paraquat. Now, I didn't have time to discuss the parallel project where we're also looking at manganese toxicity. This slide actually simply summarizes the fact that we saw the same type of impact. The overexpression of alpha-synuclein not only potentiated paraquat, but also potentiated manganese toxicity. And again, in the case of paraquat, it makes complete sense. And the target of paraquat is the PPP, again, hijacking DP6 to generate this reactive oxygen species. And what alpha-synuclein is basically doing, is the overexpression of that, again, it's blocking entry of metabolites downstream in terms of the TCA, and it's increasing the ability in terms of glucose to enter the cell. So effectively, what alpha-synuclein overexpression is doing is feeding the PPP to allow this further generation in terms of reactive oxygen species. So I'm going to go ahead and change my topic and discussion to the last topic here, the application of metabolomics for diagnosing multiple sclerosis. Like Parkinson's disease, there is no known cure for MS. It affects over 400,000 people in the US, where women are more commonly affected by MS than men. And also, this is a disease that generally hits young adults, and just like Parkinson's disease, is also progressive. One of the challenges with MS is that it's very difficult to diagnose. And it involves multiple parameters: patient's history, MRI, central spinal fluid and biomarkers, also visual evoked potentials. The result of this difficulty is that there's a really, really high false positive and false negative results. That leads to really unfortunate hallmarks that almost, on average, a patient might go over 7 years before they get a proper diagnosis, which means that they're getting the wrong treatments. And that's problematic in terms of, while there's no cure for MS, there are effective treatments to diminish the progression of the disease, but that requires early onset. And if there's delays, we get more of the serious impacts in terms of the actual disease, right? So the challenge, again, with MS is that any disease that impacts the CNS can mimic MS. And in fact, this table summarizes about 24 of well-known CNS diseases that can routinely be incorrectly diagnosed instead of MS. And so that's the challenge in terms of trying to diagnose this disease. And so multiple sclerosis really will benefit from the availability of a simple test, like looking for a biomarker, right? So along that lines, MS has been, the field has been looking for diagnostic biomarkers for the most time. That focus, understandably, has been on the analysis of the CSF, because it's going to be proximal to the myelin damage where MS starts. Of course, obtaining CSF is extremely difficult and actually dangerous. But unfortunately, to date, while there have been a number of potential markers identified, none of these have actually been successful in being useful for diagnosing MS. And again, it's well known, in particular in the field, that delayed treatment leads towards the progression of these more serious factors. And so the earlier the treatment, the better; the earlier diagnosis, the better. So there's a desperate need for this. And so when we got into this, urine had seen very little attention or investigation as a source of MS diagnostic biomarkers. And in fact, I still think people question in terms of the utility of this, since the impression is actually downstream from the actual disease, taking place in terms of the myelin sheet. But clearly, there's a huge advantage in terms of using urine versus CSF, because it's non-invasive, it's safe, it's cost-effective. Patients can actually collect their samples themselves. So we went ahead and, despite the perspective of CSF being the proper choice, we decided to go ahead and explore the utility of using urine. And there's a well-established animal model for MS. It's called the experimental allergic encephalitis, or EAE mouse model. Again, it's using female C57B6 mice. And if you treat them with an antigen over about a 11 to 14 day period, they develop symptoms that are very reminiscent in terms of MS. And these symptoms can also be very clinically scored based on well-established appearance. So this model was developed back in the 1930s. So we went ahead and used that model to collect urine samples from healthy mice, diseased mice, and also mice receiving a treatment in terms of MS. And we initially are doing this in terms of using NMR. And even if you're not an NMR expert, you can clearly see that there is a very striking difference between the healthy mice and the diseased mice in terms of the metabolic profile. And excitingly, the mice that received a current MS treatment also reverted back to and resembled the actual healthy mice. All right, if we go ahead and do this in a more robust approach in terms of looking at the multivariate statistical model, using a variety of different healthy and controls mice relative to the EAE, which also include the EAE treated, we can see that in this OPS model, which again, a very highly validated model, p-value nearly 0, very high R and Q squares, that all the healthy controls and also the EAE treated mice all cluster very nicely together and are very distinct from the urine samples from the EAE mice. Again, we go ahead and use standard S-plots to identify the major metabolic differences, also looked at univariate, multivariate analysis. What we were able to actually identify is a collection of roughly about 26 metabolites that are uniquely perturbed in the urine of these EAE mice relative to healthy controls. So this at least gives us a hope that there might be a similar approach that we can go ahead and use in terms of humans. And more excitingly, if we go ahead and look at all the metabolites that were majorly perturbed in terms of the urine in the EAE mice and look at what literature values are there, we can see that they all make biological sense, that there have been previous association with MS and EA, or association with neurological problems, or again, EA are associated, previously shown to be in CSF in terms of MS patients. So again, there's across the board uniform association of all the metabolites that we were observing in the urine of mice being previously associated with MS, CSF, or EA gave us confidence that we were seeing something that was really biologically relevant. So we went ahead and did a proof of concept with some minimum set in terms of urine samples, looking at both healthy controls, MS patients, and also patients suffering from a secondary CNS disease that mimics MS. This secondary disease, neuromyelitis optic spectrum disorder, or NMOSD, actually was for a long time misdiagnosed in terms of being MS. And what we're actually able to show is that we can see a clear difference in terms of the urine samples from the MS patients from the healthy patients, and again, a very strongly validated model. And similarly, an even stronger model difference, we can see the difference between the healthy and the NMOSD patients. And again, we can go ahead and look at backscale loadings and S-plots to identify the major metabolites that are perturbed in terms of the MS patients relative to healthy or NMSOD. And we were able to go ahead and identify, again, a set of roughly about 26 metabolites that are uniquely changed in terms of MS versus healthy. And excitingly, a large number of these metabolites and pathways associated with these were also observed in the mouse model. Again, we can go ahead and take those metabolites and do a network analysis and identify the major pathways that are associated with those particular metabolites. And again, as I just said, if we compare that to what we previously saw in terms of the EA mouse model, there's roughly about a nearly 70% overlap in terms of the pathways that are affected and a 40% overlap in the exact identified metabolite between the two. So again, suggesting that the impact is similar, that's associated with the actual disease, and it's not necessarily species dependent. Again, we went ahead and looked at SUS plot loadings, comparing now MS versus the NMOSD patients. And again, we were able to observe a subset of metabolites that were specific to MS and other metabolites that were specific to NMOSD. And again, we can go ahead and look at the relative perturbation of these specific metabolites compared to both MS, NMOSD, and also healthy. And again, across the board, have statistical significance across the set of metabolites. So overend, what we saw is that we had a set of metabolites, after robust false discovery and secondary statistical analysis, we were able to observe a set of metabolites associated with MS. And also we were able to observe a subset of these metabolites that differentiate MS from NMOSD. Right? And again, I want to stress the point that these subset of metabolites went through extensive corrections to determine statistical significance. So another thing that we're interested in terms of this particular project, can we now also use these metabolite biomarkers as a means to monitor a patient's response to treatment and progression associated with the actual disease? So we collected another set of urine samples from human patients before in terms of naive treatment or after 12 months in terms of receiving a dimethyl fumarate DMF treatment. We were able to go ahead and do a multivariate ROC analysis, and we were able to again identify a set of 5 metabolites that differ significantly between the treatment naive and the DMF treatment. So in other words, we can see a patient's response to the treatment before they started the treatment, at the very beginning in terms of the diagnosis, in terms of MS, and then afterwards, right? And again, univariate analysis confirmed these sets of particular metabolites, right? So where are we now in terms of this project? This project is still ongoing, and we have expanded our data set. So we have over 450 new samples over all 3 different standard biofluids: urine, serum, and CSF. Again, the hope is, can we see some level in terms of uniformity across these 3, again, to strongly make that connection that the response we're seeing in terms of the urine is truly a result in terms of the pathology of MS in terms of the CSF. We have collected a variety of NMR data on these samples, again, combining NMR with MS. So to date, we've gotten well over 1,300 spectra, and our data analysis is still in progress. In this particular case, the data is predominantly driven by samples from MS progressive in terms of healthy controls, but we're also trying to fill in this cohort level in terms of getting additional samples from our local collaborator. Furthermore, if we're going to go ahead and utilize metabolite biomarkers as a means for diagnostics using NMR or mass spec, might be a very expensive and costly approach. So one of the things that we have been looking at is the idea in terms of using a microfluidic device. And this is actually a relatively simple system. It's a literally printed paper that's then laminated. We can go ahead and apply our small amount of our sample, a few microliters, and within a few seconds, the 8 different wells can be filled up. And we can associate, and again, this is simply a proof of principle using traditional detection in terms of vitamin C or caffeine. We can see a color response in each of the detected wells versus the control wells, and we can see a titration as we go ahead and increase the concentration of vitamin C in terms of caffeine. So ideally, the concept would be we could place a small drop of urine and then have a different reaction associated with each different well to monitor any of the 8 metabolites that we have identified, and then use kind of a color wheel to go ahead and determine and differentiate between healthy versus diseased individuals. So in summary and conclusion, hopefully I've convinced you that there's a great advantage in terms of combining NMR and mass spec. Clearly improves both the coverage and reliability of metabolome. There are many sources of errors in terms of metabolomics that we have to be very aware of, conscious of. Again, particularly it deals with uses of statistics, standardized protocols, accepting the fact that the metabolome is very unstable. And we're trying to address some of these problems with the availability of our MBA-PAC toolkit. In case of the applications, we talked about and looking at the molecular mechanism of Parkinson's disease. And again, what we observed is that paraquat hijacks NADPH from the PPA to redox cycle, induce oxidative damage, and impair antioxidant defenses, which results in neurological death. Alpha-synuclein potentiates these environmental toxins by channeling carbon flux into the PPE to increase paraquat's redox cycling, and also glucose metabolism seems to be regulated by the combination of these alpha-synuclein paraquat toxicity. In the case of multiple sclerosis, we've got some very exciting, promising preliminary results in terms of identifying urine metabolites to diagnose MS, to differentiate MS from healthy and from other CNS patients, to differentiate between treatment and naive, and there seems to be some consistency across both animal and patient models. So in summary, I want to thank all my students who have worked extremely hard on this particular project. They're shown here, and some of them have actually gone on to graduate. Currently, Eli and Tao are working on further developing our MBA-PAC software, which was initially developed by Brad Worley, who's graduated. Fatima has been extremely valuable in terms of driving a lot of the methodology development, and also with the multiple sclerosis project, and also the assistant in terms of the Parkinson's disease. Also I want to thank our collaborators, particularly Rodrigo Cruz, who's been working with us in terms of the Parkinson's disease, Jay Reddy with the, and Zalt Illis with the MS project, and of course our various funding sources. And again, I'd like to thank you for your attention and participating in this webinar, and I'd be happy to answer any questions.
Okay, I'm back. So we have a few questions in the questions board. Maybe we can start with that, and I'll give the rest of you a little bit of time if you have any more questions. Please go ahead and type them out in the questions section. And if your question is answered and you still have a follow-up, please do so too. I guess we'll start backwards. The last question on this from Sachet Shukla is, have you checked the MS signature metabolites or MS signature in blood? And another question is it possible to infer a metabolic signature in Parkinson's disease for early detection, or if there's an effect of treatment based on pre and post metabolic profile? So it sort of ties into how early detection shown in MS can be sort of used for PD, and if in MS there is a value to using blood as the metabolic signature.
So let me answer the blood question. I would think it would be equally valuable. It's obviously a little bit more difficult to acquire blood. Obviously requires a needle prick, and then it requires a little bit more of processing of the sample, because you need to go ahead and remove all the blood cells to get down to the serum in terms of doing the analysis. But I think that it ties into the logic in terms of why we think we've been successful in terms of using the urine, is that the body maintains chemical homeostasis, and so at the locations in terms of where the damage is taking place, at the neuron level, at the myelin sheets, those degradation products are going to be cleared. They're going to be moved into the blood and then eventually extracted into the urine. And And I think that's the logic that we're applying. So if there's value in terms of the urine, there also would be value in terms of the serum or blood, and that's also why we're going ahead and look at that. And like I said, the only advantage is simplicity. If you can see the same information or get the same value out of the serum versus the urine, then why not just do the easier route and do the urine? So Fatima, can you repeat what the question was against in terms of with Parkinson's disease? I think it was related to differentiating between disease biomarkers and response to treatments.
Yes, so it says, is it possible to infer a metabolic signature for Parkinson's disease in the context of early detection or an effect of treatment based on pre and post metabolic profile? So kind of what you'd explain for the MS project is if you could do that for Parkinson's disease, since there is such a tremendous impact on the cells pre-post treatment, and would it help in a way to develop markers for early detection?
So I think the answer to that is a clear yes. I think similar things that we've been observing with multiple sclerosis could be in fact implied to Parkinson's disease. I think one of the challenges though is it would be getting biological samples associated with very, very early symptoms before the symptom starts manifesting, because that's usually how I think most patients realize that they're dealing with chronic illness, is that they start having tremors or loss of muscle control. But yes, I don't see any reason why the same idea, the same approach that we've been doing with multiple sclerosis could not be applied to Parkinson's disease.
All right, moving to the first half of the presentation, there's a couple questions. One question says, how about using either NMR or MS to detect the metabolites in a targeted way? And in that case, when you use just targeted metabolites, there will be no noise in the data, and thus solving or helping the overfitting problem.
So yeah, I think that both untargeted and targeted metabolomics have equal value. There's clearly advantages to both approaches, and similarly in terms of disadvantages. And I think you did highlight some of the advantages in terms of the targeted approach. I guess the way to think about it, in order to induce the targeted approach, there's a lot more work that has to be done up front to calibrate and standardize the protocols to detect the subset of metabolites that you want to see. But then after those are established, then there is a more efficient means in terms of analyzing the data. Whereas when you're dealing with untargeted metabolomics, a lot of that work is now done at the back end to characterize and identify the particular metabolites. That's where a lot of the time and resources need to be embarked in. So yeah, I see value. I think both targeted and untargeted are going to be equally contributors to a number of these particular projects. Targeted makes a lot more sense when you're dealing with systems that you have some prior knowledge, so you know which specific metabolites or lipids that you want to explore. Untargeted, the strength of that approach is a discovery. You don't know what you're looking for, and so you're going to try to measure and detect everything that you can.
Another question says, what do you think about using PCA or other multivariate analysis tools only for data overview and summary, and then relying on univariate analysis for metabolite level or specifics, and then how that sort of strategy would be more relevant in the clinical setting?
So in our group, we do that. We use PCA as kind of our initial overview. There's a lot of value to that, especially since it's an unsupervised technique. So you know that if you see a difference in terms of your groups, that there is some difference there to explore. It's important to be careful though, in that PCA only identifies the largest variance in your data sets, and that variance might not be what you're looking for. You know, for instance, if you're looking for the difference in between MS patients and healthy, you might see a very large variation in terms of the PCA, but it might not be due to the underlying disease. It could be due to some other confounding factors. So I think following up a PCA analysis with an OPLS to further verify the fact that you're seeing group separation associated with what you're identifying in the group is also a valuable approach. And then, like you said, I think it would be perfectly fine to go back in terms of looking at the univariate analysis. But again, it's important to appreciate that univariate and multivariate analysis are asking different questions, and they're going to get different responses. So the univariate analysis, again, is going to be done independent of the multivariate analysis. So you don't know specifically if the variance in a specific metabolite is associated with, you know, the biological system you're asking for, or just happens to be some secondary confounding effect. So I think it's important to generate some connection between both the univariate and the multivariate analysis to know that the metabolite is associated with the actual specific disease state. But yes, we use them both, and I think that's what the field should be doing.
All right, we'll do one last technical question from the first part, and it says, what software do you use to simulate the NMR spectra?
Well, actually, the grad student who's in our group, Tao Vu, is actually getting a PhD in statistics. She developed the software in-house to go ahead and do that, to simulate that. And she's further working on and developing that to help us with our work and our research. So hoping to apply that to both 2D data and also to use that as a means to facilitate an automated or semi-automated metabolite assignments.
So MVA-PAC can help simulate NMR spectra too, or is that a plan in the future?
Yeah, it's currently not part of MVA-PAC at this point, but that would be a plan in the future, yes.
Okay, I think this is an interesting question, and I think we'll end with this one. Have you thought of metabolite network approaches to expand the network of compounds known and unknown that can be related to different conditions, which may not be detected using a univariate, multivariate analysis approach?
So I agree, Fatima, that is...
Yeah, I'd like if they would expand on what they mean by the metabolite network approaches and what sort of algorithms are part of that. But yeah, that's where we're moving, but I'll let you answer it.
Yeah, well, like I was saying, I think that's a great question. And yeah, I completely agree that that's the future of the field, be able to combine what we can observe experimentally with robust predicted models. And I think there are a number of ways that people are thinking about doing this in terms of how do you simulate the entire metabolic network or associated pathways within a cell, and then use the experimental data as a means to monitor where perturbations occur, and then leverage that to expand beyond what's detected to the other pathways and metabolites, exactly as said. I think the challenge is still limiting in terms of what we know and don't know. And so while we know a lot of the pathways and we know a lot of the enzymes that are involved, there's still a lot that we don't know. And in order to be able to go ahead and start simulating reliably and accurately from a computational approach, we also need experimental parameters and measurements. What is the equilibrium constants and kinetics and turnover rates of each particular enzyme, so you can make up a correct analysis of that. So a lot of that is not known, and so when people are trying to make these models, they're having to infer or predict those values. And what you observe is that it doesn't take very small change in an equilibrium constant or enzymes turnover rate to lead to very large perturbations in terms of downstreams. So the answer is yes, that's where we need to go. That's going to be the only way for us to uncover, the rest of the dark metabolome. Combining NMR with mass spec and with IR and other analytical techniques is only going to get us so far. The metabolites still have to be at some level of detection, and a lot of these are going to be very quick turnovers. They might not exist enough for us to be able to go ahead and detect.
Okay, I think this is very informative and very detailed. I think all of the listeners, especially the early career members, hopefully you can incorporate some of these suggestions into your future work. We will not have a webinar next month in June. We will continue back up in July. So you will get our emails as you do if you're subscribed to the Metabolomics Society email listserv. Thank you, everyone. And we will have the video of the webinar posted once we've like converted everything and ready to go. If you would like a reminder about when the video is updated, please let me know either in the chat or something, and I can send you a specific email. I will try to send an email blast to everyone as well. So thank you, everyone. Thank you, Dr. Powers, for taking the time out to do this webinar. We really appreciate it.
Well, thank you, and you're welcome.